Hello. How do organisms survive freezing temperatures? This is something we will discuss in the following two lectures. Although a few flying insects, for example the monarch butterfly, are actually migrating to warmer regions during winters, the majority of ectotherms must somehow deal with freezing temperatures for shorter or longer periods. Most organisms try to avoid the most extreme low temperatures by hiding below the snow surface or even move to sheltered habitats deeper in the ground. As seen in this graph, soil temperature is much buffered by snow or litter and generally is much warmer than air temperatures. Despite these behavioral strategies, many organisms will be exposed to sub-zero temperatures during winter. These organisms must resort to physiological adaptations that enable them to tolerate low temperatures. To begin, I'll introduce some important definitions that are related to the cold tolerance of ectothermic organisms. For these organisms, cold tolerance is very much a matter of avoiding ice formation in their body or tolerating ice formation. Freeze tolerance is the ability to tolerate ice crystals in the extracellular compartments of the body. Ice formation in the cell's cytosol is rarely tolerated. This is remarkable as a strategy and has evolved in many different ectotherms, including plants. Freeze intolerance is obviously the opposite and is a characteristic of the freeze-avoiding organisms. So these animals are seeking to avoid internal ice formation by various adaptations that we will look at later on. Since cold tolerance is very much related to the physical chemistry of water, it's important to be acquainted with the meaning of melting and freezing points of aqueous solutions such as hemolymph. If you imagine an apparatus in which you can closely control the temperature of a small volume of hemolymph, you can freeze such a sample and then slowly heat the sample while you observe the ice crystal. The melting point of this sample is defined as the temperature where the last tiny ice crystal will melt while heating. In artificial aqueous solutions, the ice crystal will immediately grow when the sample is cooled. This is the freezing point. However, in many biological solutions, the melting and freezing points are not always the same. The melting point of a solution is closely related to the concentration of solutes. The osmolality of a solution is a measure of the total number of osmotically active solutes. We can convert osmolality to melting point using the osmolar melting point depression constant, which is minus 1.86 degrees centigrade per osmol. The last important definition to be introduced here is the supercooling point, or temperature of crystallization. This is the temperature where a volume of, for example, hemolymph spontaneously freezes when it's cooled below the melting point. This phenomenon, supercooling, is utilized by many freeze-avoiding species, making it possible to avoid ice formation even though the temperature is much lower than the melting point of their body fluids. As outlined in the paper by Sinclair and co-workers, the cold tolerance strategy of an organism can be determined by investigations of survival after exposure to a range of sub-zero temperatures and coupled with measurements of the average supercooling point of a species. If we measure the body temperature of an organism and expose it to gradually decreasing temperature, we can observe the supercooling point and from that deduce if it uses freeze tolerance or freeze avoidance as a cold tolerance strategy. Species that do not survive even modest low temperatures, slightly below the melting point of its body fluid, are termed chilling intolerant. This is characteristic for many tropical or subtropical species, but also many species living in temperate regions. For example, many fruit flies are chilling intolerant and will die from a few hours of exposure to moderately low temperature. Species that survive in a supercool state for extended periods, that will be days, weeks, or months, at temperatures below their melting point but above their supercooling point, are termed cold tolerant but freeze avoiding. And once the temperature reaches the supercooling point, liquid water crystallizes and turns into ice. During this phase transition, latent heat of crystallization is released, and this can be detected by, for example, a tiny thermocouple attached to the organism as you see to the right. Organisms that tolerate freezing and internal ice formation for longer periods, again days, weeks, or months, are termed freeze-tolerant organisms. One of the most important effects of low temperature is the risk of phase transitions in the cell membrane. At normal non-stressing temperature, the composition of phospholipids ensures that the membrane has the right fluidity for being fully functional. This state is called the liquid crystalline phase. When temperature is lowered, the membrane will gradually become less fluid and will eventually go into a gel phase where the phospholipid fatty acid chains are packed more closely. This has a negative impact on the functioning of integral proteins such as the sodium-potassium pump, which maintain a proper polarization of the intra- and extracellular compartments necessary for cellular functioning. At low temperature, the active pumping of potassium out of the cell cannot keep pace with the passive influx of potassium across the cell membrane, and this causes chill injury that develops because cellular depolarization occurs. The depolarization-induced injury is further enforced by opening of voltage-sensitive calcium channels, causing an influx of calcium, which gives a calcium overload that triggers apoptotic and necrotic pathways and cell death over time. A very important adaptation is therefore the ability to restructure and tailor the composition of phospholipids of the cell membrane to ensure the right fluidity at low temperature. Membrane fluidity is mostly determined by the nature of the polar head group of the phospholipid molecule, the number of double bonds in the fatty acid chains, and the length of the fatty acid chains, as you see to the right. Organisms have very efficient mechanisms to either introduce more double bonds in the existing fatty acid chains or de novo synthesize phospholipids that have more double bonds and shorter fatty acid chains. This process is termed homeoviscous adaptation. Many comparative studies have shown that within a species, membrane fluidity correlates with cold tolerance. This has also been observed in laboratory acclimation studies or in comparative studies of populations that are collected along, for example, a climatic gradient. Whereas low temperature in itself can cause injuries, freezing of body fluids also has dramatic effects on cellular homeostasis. On the first picture you see the normal situation where extra and intracellular concentration of solutes, these are the small dots on the figure, is the same. At temperatures below the melting point of body fluids, there's a risk of freezing and ice formation, which will almost exclusively occur in the extracellular compartments. Ice crystals are formed purely by water molecules, and this will cause the concentration of solutes to increase, as you can see in the middle figure. When this happens, water molecules will be drawn out of the cells until there is equilibrium between extra and intracellular osmolality. So this causes cells to shrink, and the lower the temperature is, the more the cells will become dehydrated. The cell shrinkage can eventually cause membrane fusion, and further, very high concentrations of salts can result in protein denaturation and precipitation. So these were some fundamental considerations of cold stress. We now turn to describe and discuss some major cold tolerance strategies, the first one being freeze avoidance. Freeze-avoidant species rely on their ability to remain in a supercool state. Staying unfrozen at sub-zero temperature is very much a matter of chance. The risk of freezing increases with lowering of temperature, but also with increasing exposure time. So sooner or later a frozen or a supercooled sample will actually freeze if we wait long enough. If we take the example of this hibernating larvae of this wheat stem sawfly, we see that the time until freezing is very short at low temperature, actually only a few minutes. However, at higher sub-zero temperatures, about -20 degrees centigrade, this larvae can remain unfrozen and supercooled for weeks or months. In large animals, there are many more ice nuclei that can initiate the freezing. Therefore, small organisms are often better supercoolers than large organisms. The rather large wingless Orthopteran, the New Zealand weta, as you see here, it actually weighs about 10 grams, has a supercooling point of -5 degrees centigrade, while the springtails, weighing only 1 milligram, have supercooling points below -15. We will now look at important adaptations of cold-hardy species that use freeze avoidance as an overwintering strategy. A very efficient way for these organisms to improve supercooling ability is to get rid of all so-called ice-nucleating agents. Food particles in the gut can initiate freezing, and animals typically stop feeding and clear their gut when temperatures in the autumn get low. For example, this Antarctic springtail will shift the supercooling point when starved, seen as a higher frequency of efficient supercoolers in panel B as compared to non-starved individuals in panel A. Animals can further improve their supercooling capacity by increasing the osmolality of their body fluids. This is typically done by accumulating low molecular weight cryoprotectants that function just like antifreeze added to the cooling system of your car engine. Glycerol is a common cryoprotectant in insects. In this example, larvae of the spruce budworm will during autumn increase the concentration of glycerol to almost 30% of hemolymph weight. At the same time, the supercooling point is lowered from -20 to -45 degrees. If we try to estimate what this glycerol accumulation will mean for the melting point of hemolymph, we find that about 270 grams of glycerol divided by the molecular weight of glycerol is equal to almost 3 mole glycerol per kilogram hemolymph. Multiplying this by the osmolar melting point depression constant results in a depression of the melting point by 5.5 degrees centigrade, and this contributes to the lowering of the supercooling point. Supercooling capacity of Arctic insects are remarkable and very good examples of how a species can be adapted to very low winter temperatures. For example, these midges from the interior Alaska can stay supercooled at temperatures below -40 degrees for several months during the Arctic winter and will survive the winter in this state. Desiccating conditions in the environment can cause organisms to dehydrate. Thereby, the osmolality will increase, and that lowers, as we saw before, both the melting point of the body fluids, but also the supercooling point. This example with an Arctic springtail shows how the supercooling point of an animal decreases as the animal gradually loses body water. The body water content is therefore very important for the survival in a supercooled state. This mechanism is of course only adaptive if the organism can survive considerable dehydration. Inoculative freezing describes the penetration of external ice spicules through the openings in the skin or cuticle of an organism, or even through the mouth or anus, which thereby seed freezing of the internal body fluids. The wax layers of the insect cuticle is an efficient barrier for inoculative freezing. As seen in this example, removal of the waxes and lipids of the epicuticle, actually by washing with a detergent, will greatly increase the risk of inoculative freezing. Inoculative freezing is especially important for animals overwintering in soil because they are in close contact with ice crystals when soils are frozen. In this example, you see that water content of soil has a large impact on inoculative freezing and therefore also mortality. The higher the soil moisture level, the more ice crystals will be in contact with these potato beetles, and therefore the higher the risk of inoculative freezing. Since this species is freezing intolerant, mortality is directly linked to the proportion of frozen beetles. In the next presentation, we'll go and look at another strategy, the freeze tolerance.